Assalamu alaikum. So let's continue talking about regulation of enzymes. So previously, in the previous lecture, we talked about expression of isoenzymes and how they are regulated and how they differ kinetically. And we also talked about inhibitors and what they do and the different types of inhibitors. Now, in this lecture, we're going to talk about conformational changes and how enzyme activity is, or it can be controlled by changing their structure. So let's talk about the first part, rate of regulating enzyme activity via conformational. So let me introduce first the metabolism of glucose. So we said that the first reaction in the glycolytic pathway is phosphorylation of glucose into glucose 6-phosphate. Now we can go on in 9 different reactions to break down glucose 6-phosphate into 2 molecules of pyruvate. This is known as glycolysis. Now another fate for glucose 6-phosphate is to convert it into, or to add it into glycogen, so increasing the number of glucose residues in a glycogen molecule. Okay, so this is known as glycogenesis, okay, creation of a glycogen molecule. Now similarly, whenever the body needs glucose, glycogen, which mainly exists of course in the liver and to some extent in muscles, glycogen can be broken up into glucose 6-phosphate, which can then be converted to glucose, and glucose is transported out of liver into peripheral tissues. This is known as glycogenolysis. Okay? Now let's zoom into the formation and breakdown of glycogen molecules. So we have a glucose 6-phosphate. This is converted to glucose 1-phosphate. So these two are isomers. Now glucose 1-phosphate is converted to UDP glucose, so the phosphate is removed. UDP is added to glucose, making it a more active molecule that can be used to integrate it into a glycogen molecule via an enzyme known as glycogen synthase. Okay, so UDP is released, and the energy that is generated from the release of UDP is used to integrate this glucose molecule into a glycogen. Now we have a glycogen molecule that is larger by 1 glucose molecule. So whenever the body needs glucose, glycogen is broken up, or let's say 1 glucose molecule is removed from this glycogen molecule enzymatically as well via an enzyme known as glycogen phosphorylase. So this glycogen phosphorylase uses a molecule of inorganic phosphate releasing a glucose 1-phosphate, or releasing glucose in the form of glucose 1-phosphate, and now glycogen is less by 1 glucose residue. Now glucose 1-phosphate is converted to glucose 6-phosphate, and so on. Okay, so we're going to focus on 2 enzymes in here. The first is glycogen synthase, but mainly we'll focus on glycogen phosphorylase. Now these 2 enzymes, the glycogen phosphorylase and glycogen synthase, are regulated simultaneously by a single enzyme known as protein kinase A. Okay, so the idea here is that with one hit, by activating protein kinase A, you turn off the glycogen synthesis pathway, and the glycogen breakdown is turned on by glycogen phosphorylase. And that makes sense. This is quite efficient because these two reactions should not be active at the same time and should not be inactive at the same time. One should be active and the other should be inactive. Okay, so by a single stroke you have this happening. So we have this enzyme right here, that is protein kinase A. Now this enzyme is actually regulated at the hormonal level. So whenever the body needs energy, whenever there is low level of glucose in blood and peripheral tissues need blood, it's time for liver to break up or break down glycogen into single glucose molecules that can be transported to peripheral tissues. Okay, so you have these two hormones, epinephrine, which acts on muscle cells, glucagon, which acts on liver cells, activating protein kinase A, and protein kinase A does the rest. Now, so let's talk about protein kinase A. Now, this is a multimeric kinase. It is a serine threonine protein kinase, meaning that it phosphorylates, that is, it adds a phosphate group on serine and threonine residues in other proteins. So what this enzyme does is that it can phosphorylate many proteins. It adds phosphate groups to many proteins, activating them or inactivating them. So this is, as I said, a multimeric protein, meaning that it has a quaternary structure, right? It is composed of 4 polypeptide subunits. 2 of these are known as regulatory subunits, and the other 2 are known as catalytic subunits. So these 2 catalytic subunits catalyze the reaction of phosphorylation, and these 2 regulatory subunits regulate the catalytic activity of these 2 subunits. So what happens with protein kinase A is that it can act on many other kinases, okay, activating them or inactivating them, as I said. Now, this is a master enzyme because it can act on many molecules. So it can influence the activity or the signaling of many pathways. Okay, so of course this is not for you to memorize, but just to show you how important this protein kinase A is. And it can be influenced by many pathways as well. Okay, so they all converge on protein kinase A, and protein kinase A can influence the activity of many other enzymes. So this is really a master molecule. Now, so how is cyclic AMP affected? How is it regulated? It's rather regulated by a small molecule known as cyclic AMP. Okay, so this cyclic AMP is produced from ATP. So ATP is adenosine triphosphate. We introduced ATP and the structure of ATP in the molecular biology course, if you remember. So right here we have the adenine nitrogenous base. We have a sugar, ribose sugar, and 3 phosphate. So this is adenosine. These 2 are they constitute adenosine triphosphate. Now this ATP is converted to cyclic AMP because you have the formation of a cycle right here by an enzyme known as adenylyl cyclase. Adenylyl cyclase. Okay. Now this cyclic AMP influences the activity of protein kinase A. In order to remove cyclic AMP and to terminate its action on protein kinase A as well as other enzymes, it gets converted to AMP, adenosine monophosphate, by an enzyme known as cyclic AMP phosphodiesterase. Now, what happens with cyclic AMP is that it binds, two molecules bind to each regulatory subunit. This binding results in change of the conformation, the structure of the regulatory subunits, and the catalytic subunits become free. Now, each one of these subunits is very active, and it starts to modify other enzymes and other molecules, influencing their actions. So you have disruption of the quaternary structure of the protein kinase A. Okay? So, basically what happens is that you have a hormone that binds to a receptor, and this receptor activates adenylyl cyclase. Adenylyl cyclase can then form cyclic AMP. Cyclic AMP binds to protein kinase A, activating it. So let's talk about reversible covalent modification now, which is another form of regulating enzyme activity. So basically, we already talked about phosphorylation. That is the addition of a phosphate group to proteins. Okay, that's what protein kinase A does. It phosphorylates amino acid residues in proteins and enzymes. So this phosphorylation, the addition of a phosphate group, can be done on amino acid residues, mainly serine, threonine, as well as tyrosine. So you have what is known as serine threonine kinases. They phosphorylate serine and threonine residues, and you have tyrosine kinases as well that phosphorylate tyrosine residues. Now, the advantage of phosphorylation is that it is rapid and transient, meaning that, well, it takes a short time for it to happen. Transient means that phosphate group can be added and removed quickly. So usually this phosphorylation takes place by taking a phosphate group from ATP. So usually ATP is the source of this phosphate group, and these kinases take this phosphate and they add it on proteins. Okay? In order to remove these phosphate groups, we need proteins or enzymes known as phosphatases, protein phosphatases. Now remember, when we talk about these enzymes, pay attention to the classification of enzymes. So you need to know what kinases, to what class kinases belong to, and same thing with phosphatases. So this is known as phosphorylation, and the reverse reaction is known as dephosphorylation. Now note that dephosphorylation is not the reverse of phosphorylation. It's a different type of reaction. Now, addition of a phosphate group does not mean that the protein would be active or the enzyme would be active. It depends on the enzyme. So sometimes phosphorylation can turn on proteins or enzymes, and sometimes phosphorylation turns off these enzymes or other enzymes. Okay? So why is phosphorylation effective? Because what happens with addition of a phosphate group is that it can induce significant change in the structure of proteins. The reason is that the phosphate group is highly charged, negatively charged, so it does cause changes in the structure of proteins as a result of repulsive and attractive forces. Okay? Now the other thing is that it can happen very quickly. Now it can also happen over a span of hours. Okay, so there is regulation of timing of phosphorylation or dephosphorylation of proteins. Now, the other thing is that phosphorylation causes highly amplified effects. What do we mean by that? The thing is, protein kinase A can phosphorylate many molecules of kinases. Okay, so let's assume that. Now, each kinase can act on many targets. So let's say that protein kinase A phosphorylates 10 kinase molecules, and each one of them can phosphorylate 10 other molecules. So now we have 100 proteins that can be phosphorylated downstream of protein kinase A. So you have amplification effect in here. Now there are other modifications, protein modifications, in addition to phosphorylation. You can have what is known as adenylation, that is addition of an adenylyl group right here. This is the adenylyl group right here. And usually you have the addition of AMP, okay, that's this molecule right here, to tyrosine residues of proteins. You can also have the addition of uridylation, okay, same thing. Now usually, by the way, the addition of these molecules, like the large bulky molecules like AMP, would result in inhibition of enzyme activity. You can also have what is known as ATP ribosylation, the addition of adenosine diphosphate ribosyl group to inactivate enzymes as well. Now you can have the addition of small groups like methylation and acetylation, and this can also influence or regulate enzyme activity. Usually methylation is done at the carboxylic side chains of residues, okay, as well as the end of proteins, and acetylation is done via the addition of an acetyl group to lysine. Now, if you remember in molecular biology, we talked about the acetylation effect of histones of DNA. Same thing with methylation, by the way. Okay, so here's what happens. So you have protein kinase A influenced or regulated by cyclic AMP. Okay, now when it binds to cyclic AMP, it gets activated. Protein kinase A would phosphorylate glycogen phosphorylase, inactivating it, and what it does is that it phosphorylates another enzyme known as phosphorylase kinase, okay, activating it. Now remember, now this here, the name is basically, this is a kinase that phosphorylates the phosphorylase. So the phosphorylase kinase phosphorylates glycogen phosphorylase. Okay? Now, again, you have inactivation of glycogen synthesis. Now, these are the hunger hormones. Okay, so whenever we need energy, there's no need to synthesize or to enlarge glycogen. Okay, so that's the signal. So protein kinase A gets activated, it blocks glycogen synthesis, and it induces glycogen breakdown into glucose. Okay, so you have protein kinase A phosphorylating phosphorylase kinase, activating it, and this phosphorylase kinase would phosphorylate glycogen phosphorylase, which acts on glycogen to remove glucose monomers. So let's talk about glycogen phosphorylase now. Now, what this enzyme does is that it uses inorganic phosphate to remove glucose in the form of glucose 1-phosphate. So it's almost like you have this large glycogen molecule with glucose hanging down. And what the enzyme does is that it takes a rock, which is inorganic phosphate, it throws it on glycogen, and you have a glucose that comes out of it. So that's how I imagine it happening. So what glycogen phosphorylase does is that it catalyzes removal of glucose molecules from glycogen, and you have a serine residue that gets phosphorylated, and it is away from where the active site is, but it influences the structure or the shape of the active site. Okay, now this glycogen phosphorylase exists in 4 forms. Okay, so pay attention. You have what is known as a T form, and you have an R form. T form means that the enzyme is not active. The R form means that the enzyme is active. Okay, just like hemoglobin. That's one. Now the other 2 forms is what is known as phosphorylase B and phosphorylase A. The difference between phosphorylase A and B is that phosphorylase A is phosphorylated and phosphorylase B is dephosphorylated. It doesn't have a phosphate group. Okay? So you have phosphorylase B and phosphorylase A. The difference is that one is phosphorylated, which is A, and the other is not, which is B. Now both phosphorylase A and phosphorylase B exist in 2 forms, 2 structural forms, R form and T form. So this is an allosteric enzyme, just like hemoglobin. Now note the arrows in here. When the enzyme is dephosphorylated, it mainly prefers to be in the T state. So you have phosphorylase B in the T state. That's the inactive state. Now, when the enzyme is phosphorylated, it prefers to be in the R state. So phosphorylase A mainly exists in the R state, in the active state, and phosphorylase B mainly exists in the T state, that is the inactive state. Now, the question is, can you have phosphorylase B, which is dephosphorylated, in the R state? Can you have it active? Yes, you can. Okay? But mainly it is in the T state. Can you have a phosphorylated enzyme that is inactive? And the answer is yes, you can have it in the T state. Okay? But mainly when phosphorylated, phosphorylase A prefers to be in the R state. Now, so what happens here again is that in order to activate the enzyme, this phosphorylase, it gets phosphorylated, okay, by phosphorylase kinase. Okay, so what happens is protein kinase A is activated, it phosphorylates phosphorylase kinase, phosphorylase kinase phosphorylates glycogen phosphorylase, okay, and you have the equilibrium going towards the formation of the enzyme in the R state. Now on the other hand, by removing this phosphate group, what happens is now the enzyme is dephosphorylated, which prefers to stay in the T state. Now, the transition from the T to the R state is regulated by the energy charge. This is particularly true for phosphorylase B. Okay, so when the enzyme is dephosphorylated in the presence of ATP and glucose 6-phosphate. This means that there is a lot of energy in the system, which means that there is no need to break up or break down glycogen. There is no need to remove glucose from glycogen, so because there is enough energy, whether it is in the form of ATP or if there is a lot of glucose 6-phosphate in the system. So what these two do is that they bind to phosphorylase B and they switch the conformation from the R to the T state. So these are inhibitors. On the other hand, if the system needs energy, and this is signaled by AMP, AMP binds to phosphorylase B and it switches the conformation to the R state. And this is true only for phosphorylase B, not phosphorylase A. Okay? So, the other thing is that phosphorylase A can be inhibited. It can be transformed from the R state to the T state by glucose. So there is enough glucose in the system. There is no need to break down glycogen into glucose monomers. So the phosphorylase is inactivated by glucose. Now, what ATP and AMP do is that they bind to what is known as nucleotide binding sites within the enzyme. Okay, so there are certain regulatory sites, okay, remember that, whereby ATP and AMP can bind to, and they change the conformation of the enzyme from the T to the R state. So what happens is that AMP would bind to phosphorylase B, switching the conformation from the T to the R state. Okay, so AMP signals lack of energy in the system. Phosphorylase becomes active, and it becomes in the R state, and it starts to remove glucose monomers from glycogen. On the other hand, when ATP binds to the nucleotide binding sites, the structure switches from the R state to the T state. There is no need to break down glycogen into glucose monomers. Now remember that glucose 6-phosphate also favors the T state of phosphorylase B. Okay, now so we have two modes of regulation here. The first one is via AMP. So AMP binds to glycogen phosphorylase B, again switching the molecule to the R state, to the active state. And the other mode is phosphorylation by phosphorylase kinase, and when phosphorylated, the enzyme mainly switches to the becomes phosphorylase A, which prefers to be in the R state as well. So what we have just talked about is how allosteric enzymes can be regulated. So mainly phosphorylation. So let's talk about another mode of regulation of proteins and enzymes. Now we're going to talk about large modulators, and these are known as trimeric large G proteins. Okay, so these proteins are composed of 3 subunits, and that's why they're known as trimeric: alpha, beta, and gamma. Now this G protein is usually associated with what is known as G protein coupled receptor, GPCR. Okay, so you have a receptor on the cell surface, and associated with it you have a G protein. Now the ligand or the agonist comes in, it binds to the receptor. The receptor changes structures. This results in release of GDP. Now note that the alpha subunit is associated with GDP. When the receptor changes structures, now this induces the release of GDP, and GTP comes in, the guanosine triphosphate comes in, and it binds to the alpha subunit. So this is guanosine triphosphate. Now when GTP binds to the alpha subunit is released from beta and gamma. Okay? So, and it becomes active. So beta and gamma are inhibitory subunits of alpha. Now alpha is an active molecule. Now alpha is not an enzyme, but what it does is that it binds to enzymes, okay, modulating them, influencing them, activating them. One of these enzymes is adenylyl cyclase. Now in vision, and we're going to talk about visual transduction, that is signaling, the alpha subunit is actually an activator of phosphodiesterase, which degrades cyclic AMP. So overall, what happens is that the alpha subunit is an activator, and it's a large modulator. Okay, so it's larger than cyclic AMP. So what the alpha subunit does is that it binds to receptors, it binds to enzymes, activating them. Okay, now beta and gamma subunits can also bind to other effectors as well in some systems. Now what's important here is that also another point is that the alpha subunit must be inactivated, and it gets inactivated by hydrolyzing, by hydrolyzing GTP into GDP, the diphosphate. And when GDP is associated with the alpha subunit, now alpha subunit can bind to beta and gamma, it gets inactivated, okay, and it waits for another signaling that comes from an agonist or a ligand that binds to a G protein coupled receptor. So there are other molecules like these large trimeric G proteins, and these are known as small monomeric G proteins because they are composed of a single polypeptide chain. So they do not have a quaternary structure. Now what happens with these proteins, and they include RAS, Rho, RAB, other proteins, and we already covered some of these proteins in the molecular biology course and the cell biology course. So these proteins are usually associated with guanosine diphosphate, and they are inactive. What happens is that you have removal of GDP, okay, and association of GTP. To tell you the truth, I don't like this arrow in here because that means that this GTP is converted to GDP, which is not right. What happens if you look at in the previous slide, you have GDP coming out, and then another molecule of GTP coming in. So regardless, so what happens here is that you have exchange. GTP replaces GDP. Now when these proteins, the monomeric, the small monomeric G proteins, when they are bound to GTP, now they are active. And just like the alpha subunit in G proteins, these small monomeric G proteins, when they are bound to GTP, they can bind to enzymes like RAF, or MAP kinase, the MAP kinase pathway, activating this pathway, signaling pathway. In order to inactivate, to terminate this signaling pathway, GTP is hydrolyzed, this just like the alpha subunit, into GDP, and it becomes inactive. So these proteins, just like the large trimeric G proteins, they are regulated by binding to these nucleotides, the GTP or GDP. Now these the exchange of GDP into GTP and the hydrolysis of GTP to GDP, both of these are facilitated by other regulatory proteins, including what is known as GTPase activating proteins, guanine nucleotide exchange factors, and GDP dissociation inhibitors. So what do these do? Now what happens with the guanine exchange factors is that they bind to the monomeric proteins and they facilitate the release of GDP and binding of GTP instead, activating these proteins. Now the gaps are activators of intrinsic enzymatic activity. Intrinsic means that it means that the enzymatic activity acts on the protein itself, not any other protein outside. So you have a GTPase activity, and what this does is that it hydrolyzes GTP into GDP by removal of a phosphate group. Okay, so what gaps do is that they activate the GTPase intrinsic activity, inactivating the monomeric proteins. Now the final regulators are known as GDP dissociation inhibitors. So what these do is that they fix GDP onto the protein, keeping the protein inactive. So we have two inhibitors and one activator. Finally, we're going to talk about the last mode of regulation, which is irreversible covalent modification. Now, pay attention that the previous mechanisms are reversible. So whether we're talking about phosphorylation, adenylation, or whether we're talking about GDP exchange, these are all reversible. Now we're going to talk about irreversible covalent modification. Now, enzymes that require irreversible modification are known as Zymogens, and these Zymogens are also known as pro-enzymes. Okay, the pro means that the enzyme is inactive. It's a precursor, so it requires modification, and this modification again is irreversible. So what happens here is that this irreversible modification occurs by proteolytic cleavage, okay, meaning that part of the enzyme is cleaved off, and usually it is the N-terminus. Okay, so few amino acids at the N-terminus are removed. Now, usually what happens with these enzymes is that the N-terminus covers the active site, so it blocks substrate entry into the active site. When the enzyme is activated, or the enzyme is activated by cleaving off this N-terminus, this little portion of the protein, the active site becomes open and substrate can get into the active site. And these Zymogens, good examples of these Zymogens are digestive enzymes, enzymes that help us in digesting food. Okay, so usually these enzymes that include chymotrypsin, trypsin, pepsin in the stomach, and these two, the chymotrypsin and trypsin, are intestinal. So they're usually produced in the pancreas as zymogens. So you have these enzymes known as chymotrypsinogen or trypsinogen, meaning that these are zymogens. Now some enzymes like elastase, they are also produced as zymogens, and elastase is known as proelastase, okay, meaning that it's a zymogen that requires activation. When chymotrypsinogen gets activated, it becomes chymotrypsin. So chymotrypsin is the active form of the enzyme. Elastase is the active form of the enzyme as well. Proelastase means that it is in the zymogen form, in the precursor form. Now, so why is it that they exist as zymogens? Well, whenever we eat and the signal gets into the pancreas to release these zymogens, so they are released into the intestines. By the time food reaches the intestines, now these enzymes become active in the intestines. So in other words, it doesn't require, these enzymes do not require synthesis starting from the DNA level, that is transcription, then translation, activation, and so on, because it's gonna take hours and there's no time for that. Okay, food is getting down right away. Now the other thing is that they are kept as zymogens because if they are in the active form, they can degrade tissues. They act on proteins, so these are digestive enzymes. They cleave other proteins. Okay, so in order to protect tissues, they are produced as zymogens, and in order to speed up digestion, they are produced as zymogens as well.